Hello, and in yet another video, 4 students named Kylie Tan, Audra Frogoso, Stephanie Coronel, and Pauline Bucu will be modeling the stages of meiosis. Please also excuse my accent and pronunciation. Interphase. Just like in mitosis, cells go through interphase before beginning the actual process of meiosis. During this phase, DNA and chromosomes are replicated as cells grow to a mature size through undergoing the G1, S, and G2 phases of interphase. Prophase 1. In the first actual stage of meiosis, prophase 1, chromosomes condense as the nuclear membrane surrounding them breaks apart. The centrosomes also move towards opposite poles. Meanwhile, two sister chromatids are joined together by a centromere. The result is two duplicated homologous chromosomes, chromosomes that have the same sequence of genes, or tetrads. When these tetrads pair up, it is called synapsis. This doesn't occur in mitosis. During synapsis, portions of chromatids break off and attach to adjacent chromatids in a homologous chromosome. This is a process called crossing over. This allows for genetic recombination, creating variety in DNA. Metaphase 1. During metaphase 1, tetrads, pairs of homologous chromosomes, line up at the equator of the cell. Spindle fibers from one pole attach to the centromere of one chromosome, while fibers from the opposite pole attach to the other chromosome of the pair. Anaphase 1. In anaphase 1, homologous chromosomes are separated randomly towards opposite poles in the cell. The random separation is called independent assortment and results in genetic variation. Telophase 1. The nuclear membrane reforms in telophase 1 and chromosomes wind into chromatin. Cytokinesis 1. In the In the last phase of meiosis 1 cytokinesis, the cytoplasm divides. The end result of meiosis 1 is 2 haploid daughter cells. The end of meiosis 1 begins the start of meiosis 2. Hooray! Prophase 2. Chromosomes reform in prophase 2 and the nuclear membrane breaks down once again. Metaphase 2. During metaphase 2, single chromosomes line up at the equator. The centrioles form spindle fibers and connect to the centromeres this time. Anaphase 2. In this phase, centromeres divide and spindle fibers pull chromatids to opposite poles. We are now going to redraw the cells to the ease of your eyeballs. Telophase 2. You cannot see it that well, but drawn in a light green marker is the nuclear membrane reforming in each of the cells. The chromosomes then also unwind into chromatin. Cytokinesis 2. In the second cytokinesis of meiosis, the cytoplasms divide. Ultimately, the end result of meiosis 2 is 4 haploid daughter cells, each with 23 unique chromosomes. These haploid cells are the gametes necessary for sexual reproduction. Now let's review crossing over. Yippee! Tetrad. A tetrad by textbook definition is the 4 chromatids in a pair of homologous chromosomes that come together as a result of synapsis during meiosis. It is during synapsis that crossing over takes place. The textbook definition of crossing over is the exchange of genetic material between homologous chromosomes during meiosis. Can result in genetic recombination. Now let's compare and contrast meiosis and mitosis. Meiosis. The purpose of meiosis is to reduce diploid cells to haploid cells, creating gametes for sexual reproduction. In meiosis there are two cell divisions. Homologous chromosomes pair up at the equator. And lastly, the end result of meiosis is 4 haploid cells. Some things both processes share are that they both go through that PMAT stages. The nuclear membranes disappear and reappear. And both processes experience cell divisions and duplicate DNA. Mitosis. The purpose of mitosis is to produce daughter cells identical to the parent cell and helps with growth and repair. In mitosis, there is only one cell division. Only single chromosomes line up at the equator. And the end result is 2 diploid cells. Thank goodness we finally reached the end. Now for a joke. What did one cell say to his sister cell when she stepped on his toe? Ow. Mitosis. This concludes our video. We hope this was helpful to understanding meiosis. Deuces.